Hello everyone to today's MICCAI RISE Journal Club session. Today we have a special guest that I had the pleasure to meet him in the last MICCAI in Korea. His name is Nico. He's a PhD student from the ETH Zürich, and today he's going to introduce his work modeling DINO embeddings for fast and efficient unsupervised anomaly detection in medical imaging. So thank you so much, Nico. The stage is yours.
Thank you for the introduction and welcome also from my side to my talk. Um, Regarding the topic. So first of all, I wanted to tell what actually unsupervised anomaly detection is and why should we even care about. And then I will go through three different papers, one previous work and two from our lab. And then finally, I will have some final remarks in the conclusion. So why we should even care about unsupervised anomaly detection. So when a radiology scan is administered, usually there is a question behind. So there is a certain diagnosis which is in question, or just some issue that needs to be checked. And this is usually what the radiologist checks for when they are looking at the scan. But this is not the only thing they check. Also they also scan through the whole scan, and they check for anything that is out of the normal. So they check for so-called incidentalomas, so incidental anomalies, which are not necessarily important for the diagnosis itself. And for example, in cardiac MRI, this occurs in roughly every third scan that such an incidentaloma occurs, and sometimes needs to be checked as well. And so when we try to assist radiologists with task-specific models, they ideally they would help in both the diagnosis part and also finding incidentalomas. However, since models are usually task-specific, the support for incidentalomas is not really provided, or is also not the main interest. And this is exactly where unsupervised anomaly detection comes into play, because unsupervised anomaly detection actually cares about the identification of samples that deviate from the norm. And specifically in the unsupervised case, there are only normal samples used to construct a distribute or like a model of the norm. And in the medical field, usually normal samples are images from healthy patients. So in the end, we care about modeling the distribution of healthy people's images. And then during testing, we apply image which can have some anomalies or not, and the aim is to find out if there is an anomaly and also where it is located. For example, here we would like to highlight this tumor in the brain, and in the using the unsupervised setting also has two more benefits, because first of all, it avoids biases towards certain types of anomalies. So the model is not biased to detect certain diseases, and especially for rare diseases, acquiring labeled samples is usually difficult because they occur very rarely. And this is not needed in the unsupervised setting where we only learn from healthy data. Methods in unsupervised anomaly detection usually face two main challenges, or they have, to design them poses two main challenges. The first one is how should we even model the normal distribution? And the second challenge, or the second main question, is how should we, once we acquired such a model, how can we use this model to then create or to detect anomalies and create an anomaly map out of it? And maybe you can keep these 2 points in mind during the next slides, where I will go through previous work and explain the most important trends or most important lines of work for unsupervised anomaly detection. So the first line of work I wanted to talk about are self-supervised learning approaches. So there is previous work where they not only use the normal samples, but they also use normal samples with synthetic anomalies. For example, where this patch here is copied from another place, and somehow modified to blend in more or less. And then either a classification or a segmentation model is trained in a supervised manner on this normal and normal plus synthetic anomaly data. And in the so-called one-stage approaches, this model is then directly used to also, after it's trained, is also used directly to detect anomalies in real data. And then there also exists two-stage approaches, where this stage one is the training of the model, which is exactly the same. And then stage two, actually, the feature extractor is not trained anymore, and the statistics of the features extracted from all the normal images are estimated, and this distribution of the features is then used to score anomalies. Another line of work are image reconstruction methods, where a reconstruction network is trained to reconstruct only normal images. And ideally, this model then only can reconstruct normal structures and will then fail to reconstruct anomalous parts. So there should be, an anomaly should be highlighted in the reconstruction, in the deviation in the reconstructions. And these reconstruction networks can range from different generative models, such as generative adversarial networks, autoencoders, variational autoencoders, combinations thereof, or also DDPMs. And as I already mentioned before, these reconstruction networks, they should, when you feed in an image with certain pathology or an abnormal structure, ideally this gets replaced by a normal-looking anatomy. And this deviation or this replacement can be highlighted, for example, in the reconstruction error. Or other works also use the gradient of the loss with respect to the input image. Or there exists some work where the reconstructed image is iteratively restored to both look normal and also to maximize consistency between the input and the output image. And then this the deviation between this restored and the input image can be used. Similar to the image reconstruction networks, there also exists an adaptation of feature reconstruction networks, where instead of training a model to reconstruct the images or to reconstruct normal images, these models they are trained to reconstruct the intermediate features in an autoencoder. And yeah, similar as before, then anomalies should be highlighted in, or should have differences within the features of the encoder and the decoder, and can or should be found with that. Then this idea is also brought further to knowledge distillation approaches, where you have a pre-trained teacher network, where you have a student network that you train on normal images to minimize discrepancy between the teacher and the student features. So the teacher can be trained on any images, including anomalous images. However, the student only, or to the student, you only distill the knowledge of the teacher on normal images. And this approach is even brought a bit further to reverse, so-called reverse knowledge distillation setups, where the teacher and the student don't have the same architecture anymore, but they are again in an encoding-decoding setup. However, the goal is still to minimize discrepancy in the features. Then, I would say the last, last but not least, another big group of works is the feature modeling works. There, again, of a pre-trained feature extractor, the intermediate features are extracted, and the goal is to model the distribution of these intermediate features for all the normal images. And this can be done, for example, with normalizing flow models, where the somewhat complicated distribution is mapped to an easy tractable distribution using a normalizing flow model. And then during anomaly scoring, the features of the test image can be fed through the normalizing flow model as well. And dependent on where they lie on this simpler distribution space, they can be scored as either normal or anomalous. Then similarly, but a bit simpler, there is one paper where features on a patch level are extracted, and for each patch, a Gaussian distribution is fitted through the patch feature distribution. And now lastly, there are memory bank methods where the distribution of the patch of the features or of the patch features is not explicitly modeled, but all of the features are stored in a big memory bank. And this memory bank can, mainly for performance reasons, also be subsampled to have more remarkable and important prototypes. And then during anomaly scoring of a test image, the features are again extracted, and the nearest neighbor search is performed to find the closest neighbors of the test features in the memory bank. And depending on this distance, or if this distance is small, then a patch can be thought of as normal, and when this distance is big, then most likely it is anomalous. So here again, a brief overview of the 4 different groups. So we have the self-supervised learning approaches, we have the image reconstruction approaches, the feature reconstruction approaches, and those in feature modeling. And now we will dive a bit deeper into the feature modeling part. Maybe you've seen it from the abstract that we are mainly residing in this field or in this group of works. And the first approach is called Anomaly DINO, which was by other researchers published in WACV last year. And the main idea of Anomaly DINO is to use DINO features. So to use their semantic understanding of or of the semantic concepts underlying in an image. So for example, when you look at these images from hazelnuts, we have 2 normal images and 2 hazelnuts with defects. And as you can see, the defective parts here, they are highlighted in this light green. So they really stand out from what you would expect from the normal part of the shell, which is usually this purple towards olive green color. And this here is also only just a very small subset of the whole features. So in these 3 channels, this light green part seems to be similar to this part of the nutshell. However, in other dimensions, it's very different, so it shouldn't get confused with this part of the shell. So the method itself is very similar to what I explained before. So it's a memory bank approach. So they extract again DINO features from normal images and store them all in a memory bank. And the whole subsampling part is omitted. However, they control the size of the memory bank by using only a few normal reference images to extract the features from. And then during test time, features are extracted from the test image and the nearest neighbor search is performed. And just as I explained before, the distance to the nearest neighbor is then used as an anomaly score. When you look at the results, you can see that most of the anomalies are captured by Anomaly DINO. So even these small defects in the cable insulation here are captured, or even also these small cracks in the hazelnut, or also this protrusion in this pill is also captured quite well. So this means that even just training from a few or constructing the memory bank just from a few normal samples, it seems to that there is already a lot of knowledge on the normal distribution captured by that. And there are of course some limitations, and the main limitation is in the end still the size of the memory bank, because this one is proportional to the number of normal training images. And when you look at normal examples from this hazelnut dataset, there is there is a bit of variance in the orientation and the rotation. However, they look fairly similar. But when you want to use this for medical images, I visualized 4 samples from the Brett's data set, and so the variance is much bigger. You have different you have slices extracted from different positions in the brain. You have a lot of variability between different patients, and this should all be captured by the normal distribution. Otherwise, if you see a slice from a region of the brain you've never seen before, it should be treated as anomalous, and this is not what it's intended to do. And when you look at the numbers, in the paper, I think the main results were proposed for using 8 reference images. And when you use the setup from the Brett's dataset, from the BMET benchmark, there are 7,500 normal images for training. This leads to about a factor of 1,000 more features that are extracted by DINO and stored in the memory bank. And in the end, the memory bank for the industrial dataset is less than 10 megabytes in size, while for the Brett's dataset, it's about 8.4 gigabytes. And this, of course, still fits into the memory. However, when you have to look for, during testing, when you have to do this nearest neighbor search for every patch in the DINO feature map, and you have to do the nearest neighbor search with almost 6 million vectors, this is a very costly operation. And so the runtime and memory use really start to explode when you use already 7,500 images, and when you go bigger with the training data set, it gets even more extreme. And this is where we wanted, or this is a limitation we wanted to address in our next work. So we try to, in our work, we try to start again from the distribution of all the normal patch features. And the idea behind it was to find a compact and efficient model for this distribution in the feature space. And we tried to use a Dirichlet process Gaussian mixture model for that, which is somewhat indicated here. And let's parse that. So first of all, we have a Gaussian mixture model. So this means that the whole distribution should be modeled with Gaussian clusters. And the other part is a Dirichlet process, which is used as a prior for the Gaussian mixture model. And this should lead to an adaptive number of clusters. So the model should, in the end, select the number of clusters needed according to the data. So if there are more clusters needed, it has the ability to generate new clusters. And if there are too many clusters present, it can also vanish some of the clusters. And just as a little heads up, we will dive in some math in a bit. So this is how the probability of how the probability distribution looks like. So Y is the feature is a feature vector, and this is essentially a mixture of Gaussians. And the only difference at this point between the standard Gaussian mixture model distribution is that there are there is infinitely many clusters. But how this works, we will see in a bit. So the Dirichlet process consists of, or has 2 main inputs. The first one is alpha, which is the concentration parameter. And the other one is G0, which is the base distribution. You will see that in a moment. So to construct a sample from the Dirichlet process, you can, for example, use the stick breaking process. For that, we start with this stick of unit length, so of length 1. And then there is an iterative process. So first of all, we sample a VK, or V1 in this case, from the beta 1 alpha distribution. And we mark the ratio of V1 from this unit length stick. And then we break off the portion of the stick or of V1 from the stick, and we keep the remainder as well. Then we sample another V2, and we mark this ratio from the remainder of the stick. And we also break that away and keep the remainder again. And then we sample another V, break it off, and so on. And we collect all the sticks that we broke off as the pi's, which are the weights from the distribution before. So formally, the atom weights are the product of the current sample V and the remainder of all the product of all the previous remainders. And the atom itself is sampled from the space distribution. So for example, here, the atom consists for Gaussian components, it consists of the mean and the covariance matrix. And now, we can formalize this sample from the Dirichlet process as the weighted sum of the different atoms, and just with this delta distribution. So what we notice first is that G is a distribution, as it usually is when you sample from a process. And we also notice that G is discrete, so there is the probability weight is just on a countable number of points. And now back to the concentration parameter. So if alpha, the concentration parameter, is small, then we would sample from the blue dense, or this is the better distribution where we sample the V's from, so the ratio of how much we break from the stick. And if alpha is small, then we usually break off a larger chunk, or most likely we break off a larger chunk of the sticks. So we end up with fewer components, and many of the all the remaining ones just have minimal weights. However, when alpha is large, then it's most likely to only break off a smaller part of the stick. So we tend to end up with more components, or have a denser model on the theta space. And now the question is still how to use this Dirichlet process as a prior to construct our mixture model. So to do this, we can get data likelihood by integrating over theta, or by integrating the marginal likelihood times the Dirichlet process prior. And when you run through the math and put in the formula we had for G before, then you might notice from some signal processing lecture that you can take this theta k and put it into the marginal distribution of y given theta. And with this, we can take this part, the pi times p y given theta, out of the integral, and this integral then evaluates to 1. So we end up with the probability distribution we've seen before. And this is the Dirichlet process mixture model. Now, in practice, how to optimize this model, there are numerous ways, and we decided to go to optimize it with expectation maximization. And for that, the first thing to do is to be able to fit it into the memory. We had to truncate our model from infinitely a number of components to k components. But when you choose k sufficiently large, this shouldn't be a big problem. Then, as usually for EM, you construct this Q function. So here we also have this auxiliary, or the latent variable x, which is the random variable that treats the component assignment. And you maximize this for the parameters. And since we have Gaussian components, there is a set of sufficient statistics we can introduce. So for example, pk would be the sufficient statistics for the pi k. So for how likely each component, or how likely point is assigned to each component. And the same we can also define for the means and the covariance matrix. And yeah, we could use this sufficient statistics as estimates. However, we would like to not use the whole distribution of all the normal features in our memory. So we went for a batched implementation. Therefore, we introduced, or we used this exponential moving average of the sufficient statistics. And with these sufficient statistics, you can then run the math and get these two update formulas for means and covariances. these formulas, by the way, are exactly the same that you would get for a standard Gaussian mixture model. And for the stick ratios or for the component, for the component weights, it's a bit more complicated, and you would end up with this formula here. Now, initially I promised that also the number of components will be adapted to the data, and this is only possible if you also update the concentration parameter alpha. So for that, we again formulate the Q function and maximize this for alpha. And when you run through all the derivation and all the math, you end up with this update formula for alpha. And now, I wanted to show you how this evolution of fitting DPMM looks like on a little toy data set with 9 different clusters. So when you run it, there are, for example, initially it starts all a bit random, and then it fits quite soon. And for example, here on the center on the right, you can see how eventually one component goes down to the lower right, and another one arises out of more or less nowhere, thanks to the Dirichlet process prior. So now we have seen how this fitting of the DPMM works. And the next question is how we can use this model for anomaly scoring. And for this, we tried out different metrics on how the similarity can be measured between the patch features of the test image and the model we fitted before. And one metric is the likelihood of each component. One is the Euclidean distance to the closest mean, and the last one was the cosine distance to the closest mean. And in these figures, you see all the patches colored in the same color. They are those patches that correspond or that are closest to the same component. And when you compare through all these, through all the different metrics, you will find that some structures are present in some of the images. For example, this orange component here at the center line of the brain is present in this image. However, it's absent in this image here for the likelihood. But for example, for the cosine distance, it is usually more or less retained for all the images. And we observed, when we went through the data, we observed that most of the structures are predicted in the most consistent manner for cosine distance. And this is why we chose to use the cosine distance to the closest mean as our anomaly score function. So when we go back, now we also have filled in this orange box here on how to do the anomaly scoring in the end. And so now let's get to the experimental section. So we evaluated our work on the BMAT benchmark, which consists of 3 different datasets with pixel-wise annotations. And most of the results I will show you are on the Brett's dataset. And just to not overwhelm you with too many results. And in the end, after the second paper, I will also come back to the liver CT dataset as well, while the full results you can see in the papers. So first of all, let's look again at this little component visualization as similar to the one we've seen before. And here there is a little extension. We also tried our model with DINOv3, and now we can compare it. And as you can see there, the consistency of the different structures is there over over all the slices, and even for the anomalous cases. For example, ventricles here in this light blue color, they are more or less consistently assigned to the same component, even here where there are anomalies present. And likewise for DINOv3 with these gray structures. And another thing you can notice is that there are many in DINOv2 there are many of these spots where there is where there is more noise present, and this is not present in DINOv3 anymore because of a change in the model training and model architecture. And when we now look at the anomaly maps, we can see that for especially for DINOv3, for most of the or for the normal images, the anomaly scores are fairly small, and for the anomalous images, so there is a tumor here, there is one tumor here which is not that apparent, and this one here is again quite apparent. And you see that especially the one on the very right is also highlighted strongly in the anomaly maps, while the other two are a bit less easy to see in the DINOv3, and in the DINOv2 they tend to undergo in the noise. But I would argue, maybe it's biased after you've seen the image, but I would argue that you can also see the anomaly present here for sure, in the more apparent one, definitely. But let's also look at the evaluation metrics. So to do the numerical evaluation, we looked into two different metrics. The first metric is the area under the ROC curve, so under the true positive rate versus false positive rate curve. And this measures how well the model can distinguish between the different classes. So in this case, between normal and abnormal. And the other metric we used is the area under the precision recall curve. So this usually measures how pure the predicted anomalies are, or it deteriorates if there are many false positive pixels predicted. And when we first look at the area under the ROC curve, yeah, maybe before we look at the scores, I have to mention that there are with the coloring, for example, all the red colored ones are methods that rely on memory banks. So we try to group the groups we discussed initially. We group them with the same color. So we have image reconstruction, feature reconstruction, distillation, memory banks, and embedding modeling approaches. And for example, for Anomaly DINO, we have 2 points. One on the very right at high runtimes. This is Anomaly DINO evaluated or trained on the full training data set. And then on the very left, at very short runtimes, we have Anomaly DINO just with one, with the normal memory bank extracted from only one normal sample. And as you can see, our method here is with the cyan star. So we perform better in terms of AUROC compared to the 2 other methods that are in a similar level of the runtime. And we perform a bit, or there is a slight performance gap to some of the methods which require much higher runtime. But we argue that that you can make a runtime accuracy trade-off, and there it would be desirable to be as far at the top left as possible. And then when you look at the area under the precision recall curve, it shows a similar picture. However, in this time, the performance gap to the best performing method is a bit bigger. So, which means that there is still some room for improvement. And in our next work, we try to improve our model of the DINO features by using autoregressive modeling in the feature space. So previous methods, or more or less all memory bank or feature modeling approaches, they assume that the features are independent over all the pixels. So they usually estimate the probability distribution factorized over all the pixels. And the underlying assumption is that the spatial structure, which is vital for anomaly detection in medical imaging, that this structure is modeled implicitly in the DINO embeddings, or for other methods in the embedding, or in the embeddings created by the feature extractor of their choice. And we figured that when we can exploit the spatial structure of these embeddings, we could improve the model of the normal data furthermore, that we can reach better performance at smaller runtime. So our aim was to explicitly model the joint distribution of all the pixels more or less together. And for this, we try to model the joint distribution by factorizing it depending on the previous pixels in the raster scan ordering. So when you draw this in this patch grid, the patch in blue, or the probability of the patch in blue, would depend on only on the ones that it's seen previously. So in this raster scan from top left to top right, and then one column, one row down, and so on. And for our experiments, we assumed that the normal distribution, or that these conditional distributions are modeled by an isotropic Gaussian. So essentially, our model learned to predict these mean vectors dependent on the previous data. And to train the whole model, we trained to minimize the negative log likelihood, which essentially boils down to minimizing the L2 distance between the actual DINO feature and the predicted mean, plus some constant term, which doesn't matter for gradient descent. And now the question is how you can implement this in an efficient way. And there we went for an approach similar to Pixel CNN, where this factorization was proposed to be implemented in a masked convolution in a CNN. So in the first layer, operating on the DINO features, we use a kernel where only a convolution kernel where only the inputs in the white patches or white pixels is used, while in the black patches, the input is masked out. And this means, or this creates a structure that this patch here only depends on what is left and what is further up in the image. Since then, the representation of the first layer in the center of the pixel, in the center of the kernel, doesn't depend on the kernel itself. In the intermediate layers, this patch can also be, or the intermediate features of this patch can also be incorporated, as it doesn't violate the factorization we had before. And then once we have trained this model, we can simply use the negative log likelihood as an anomaly score metric. Now, when we look at the results, we can see that our method, here shown in yellow, runs very, very quickly. So even quicker than the digitally processed mixture model we ran before. And the performance in this time is also more or less on par with state-of-the-art methods, at least for the breadth dataset. For the other datasets, there is sometimes a slight performance decrease compared to Anomaly DINO. However, it performs on a very high accuracy level compared to Anomaly DINO and at a much, much smaller runtime. So this means that spatial modeling of the DINO features can improve, at least in some cases, can improve the performance over Anomaly DINO, and generally helps to train a very lightweight and efficient model. Coming to the limitations, when I showed you the datasets, I told you that we're coming back to the liver CT dataset. And results on the liver CT dataset, in terms of area under the ROC curve, they are all very high. However, when you look at the area under precision recall curve, scores are all low, which indicates that all the methods predict a lot of false positives. So this is one part where there's still a lot of room for improvement, or because none of the methods clearly work that well. Then more generally, for most or even for all of the memory bank based approaches, or even for the embedding or of the feature modeling approaches, usually the resolution of the anomaly maps stays in the same resolution as the feature maps of the model. So usually the anomaly maps are rather coarse. And another limitation is that so far no 3D information, which is, for example, present in the brain MRI brain MRI datasets, this is not incorporated at all. And this would definitely be beneficial for anomalies scoring. further down the line. So yeah, these are the main limitations I would say so far. And let's conclude. So in this talk, yeah, we looked into using DINO features for anomaly detection, and I think we can say that they are quite helpful in detecting anomalies in medical imaging. And we looked into how directly processed mixture models can be used to get a small and efficient a model for DINO features, and we that this can be used for anomaly detection. And then in the end, we also demonstrated that spatial modeling of the DINO features can be beneficial to train an efficient model for DINO features that also can be used quite well for anomaly detection. So with this, I reached the end of my talk, and I thank you all for your attention, and I'm happy to answer any of your questions.
Thank you for your presentation, Nico. It was very, very interesting. And actually, we have several questions, and we will answer all of them. The first one from Andres. What do you think about JEPA and world models approaches for medical imaging? A very general question.
Yeah, very general question. It's a tough question because yeah, I'm not super familiar with all these methods or all these models, so I think at this point it would be more or less, or I don't think I can give a very informed answer on this question.
And also, as far as I know, from previous MICCAIs, not a lot of people have tried to use JEPA and the medical imaging or at MICCAI itself. So yeah, it's an opportunity to try out and see what this model can offer. So sorry, Andres, that we cannot give you a better answer. Jay also asked, in terms of clinical utility, for example, triage of scans, which method can be fast enough to do anomaly scoring inference?
Yeah, I think for inference there are it's a bit the question on how long you can use for one sample. For example, these here are the runtimes for one 2D slice. So when you try to do anomaly scoring for a full brain MRI scan, for example, then you have about 256 scans or slices. So you would end up with about 5 seconds for one image with the models we have here. And I'm not sure. I mean, in the end, it's a question of how much how much infrastructure you're willing to set up for doing all the scorings. But I think it's still important, especially when you would like to use anomaly detection methods as a mass scanning and as you said as a maybe as a triage even. It's important to not only focus on performance but also focus on efficiency at the same time. And ideally, you shouldn't really have to choose between one or the other. Ideally, you would solve both at the same time.
Perfect. Then we with Anya, who asked, many recent methods use vector quantization and specialized code books to discretize and reconstruct representation. Do you see these approaches as functionalities similar to memory bank-based reconstruction methods and equally better?
I mean, there is, yeah, these methods, they kind of combine memory bank methods with image reconstruction methods. I I think from what I've seen, also recently, I haven't seen too many works using vector quantization or codebooks. Maybe you have seen more, Alvaro, but I think from what I've seen, they are usually also outperformed by pure diffusion-based image reconstruction methods. So I'm not sure, or there might be some room for improvements. but I think so far they're not performing better than either of these two lines of work.
I agree with that. Then we have another question, Anonymous. How do we get the certificate? I don't know what our friend referred to. Probably if you refer to the certificate of the RISE-MICCAI from the summer camp, is not here, and this was on the summer camp that happened last year, so we cannot give you a certificate. And then we have Alberto, who asked, can this DINOv3 based approach be extended to 3D brain imaging datasets?
I mean, I think you can. It's not straightforward though. I think it's still an open question on how to combine Dino features to to 3D. And I think there is a lot of work going on in this field, or also that there is more work in working on foundation models in medical imaging to extract 3D features. But I mean, here it's more or less the most it's implemented in more or less most naive way by just stacking or by training the model by slicing the images in one direction, training the model in this, for example, in axial slices for brain MRI, and then you would just stack up all the predictions. But of course, then you don't really benefit from the 3D structure.
Perfect. Then we have a question from Anis. Which features do you use from DINO? Do you only use batch features, or do you include the class token feature also?
So far, we only use the patch features, and also only at the output. I think other works for that use DINO features for segmentation. Sometimes they also use intermediate features, but so far we've only worked with the output patch features.
That's great. And Jay asked, is it just for liver CT or any CT dataset for the methods are poor? I think this referred to a previous slide that you mentioned about the CT.
Yes, about this one, but I think maybe I can go back to the datasets. Where is it? Here. I think, or I haven't checked it on other CT datasets, but I would expect that it is unique to this pre-processing of this liver CT dataset, because there are usually the foreground is, or these are samples where the foreground is somewhat large, and there are some slices where the foreground is maybe the size of this anomaly here. And sometimes also the anomalies are very tiny. And I think because of that, there is a big class imbalance, which could also affect the area under the precision recall curve. And I would argue that it's mainly the case in this liver CT dataset, but I haven't really tried it out on other CT datasets or even on other liver CT datasets.
That's okay, because you use all these experiments are from the BMAP benchmark. Yeah,
exactly. Yes,
that's okay. Another question from Jay. Are any ablations done with all the features also apart from DINO?
sorry. So with
Do you use any other
feature
extractor like a ResNet or DenseNet or something like that?
We have not. We only investigated DINO features so far. We haven't really looked into using ResNet features, but I think when you look at the results, for example, here when you look at where is the brown triangle? So patch core would be the most similar one to, or is the most similar to the pure or to anomaly DINO, just with a ResNet feature extractor.
Mhm.
But okay, it's not. It's somehow obscured behind the other ones. But when we look at the um, quickly go through here. So here for patch core, these are these points here. When you compare them with Anomaly DINO, sometimes they outperform Anomaly DINO, but I think also sometimes. So for example, for the liver CT data set, here Anomaly DINO performs a bit better than patch core. And for the retinal OCT data set, here also patch core, which is up here, it outperforms Anomaly DINO. So yeah, I think it would be an interesting step to also try out different ResNets with these models, but we haven't looked into that yet.
It's a good future work.
Exactly, yes. And I mean, there are also Dino trained ResNets, and
Yeah.
just from looking at the feature maps from those, we found that visually there is less, the features look less meaningful. But of course, for a model, it could be still more meaningful because they can capture the different dimensions better than we can because we always have to boil it down to three principal components.
Mhm. And then the final question from Alberto regarding the spatial modeling of DINO features. Did you try using attention-based or non-linear scanning-based CNN without the specialized kernel? Also, does the linear scanning require a canonical orientation and placement of the brain inside the images? Oh, wow.
So to the first part, we didn't really go much beyond the specialized kernel. What we did, maybe I can pull up this table. Sorry, it's a bit blurred. What we did is we also evaluated a bidirectional approach where we didn't only go through the raster scan direction, but we also went through the other way around. So where the factorization doesn't really hold up that well, or it's not possible anymore of the probability distribution. But even then, you don't really see an improvement or a big improvement. Sometimes there is one, but usually in the standard approach, we reached better scores than in the bidirectional approach. And I think when you try to solve it with attention-based methods, it gets hard to efficiently optimize it because now with the pixel CNN approach, you can just run it efficiently with one forward pass, while I think with attention-based methods you cannot. And there was a second part of the question. Can you repeat this
Yeah.
again?
Does the linear scanning require a canonical orientation and placement of the brain inside the images?
Um, I I don't think it requires special placement, but of course the resting or the scan direction ordering can have an effect on or can include some biases towards certain parts of the image. So because when you are at the bottom right of the image, maybe go back to the slide. When you go to patches over here, they tend to have more knowledge on what is present in the whole image, while patches up here, they have only seen background parts and not really much of the brain. So, on the other side, when we run the bi-directional model, so when we went through this way and also that way, there wasn't really a big difference. So I'm not sure, or I wouldn't argue that it's really helpful to know much more.
Okay. Also, do you try vertical or
We
or all
didn't
the
try vertical. we
or
only
other type of orientations.
no, we didn't try that.
That's a good one.
But yeah, at this point, I'm not sure if it would differ that much.
That's okay. So with this, we finished the presentation. Ilan is a great work, and Alberto, so thank you for the replies. So thank you so much, Nico, for the presentation, and yeah, I hope to see you again in MICCAI.
Yeah, thank you so much as well. And yeah, also for the audience, if you have any questions, feel free to reach out, or I'm also happy to meet you all at the hopefully the next MICCAI.
Have a
Thank you
nice
very
weekend.
much. Have a
Bye
nice
bye.
weekend. Bye bye.